So then at the end of the study, I am going to compare, so I'm comparing the response variable between the groups. You know, between the treatments. All right, that's what I'm going to do at the end. I've given everybody my treatments. I've got my response variable, maybe whether or not they recovered from cancer, did it shrink the tumor, whatever, or, you know, did my pain go down? And I'm going to say, well, what's happening over here on average? What's happening here on average? Well, we call the difference in that response variable between treatments, or between my treatment groups, statistically significant if the difference is bigger than what we'd expect to see if there's no effect. Okay, recall that when we take samples, all right, so we expect the average or proportion of different samples to vary from each other. And that is called sampling error. All right, so if I took identical, if I randomly picked 100 students and looked at their average GPA, and then I did a completely other sample, so I'm going to randomly pick 100 other people and look at their GPA, there's no reason for them to be different, but they're going to be different by a little bit. All right, so data varies. We expect that. But if the difference, now, I mean, obviously those are going to be close to each other, and I expect some sort of random variation. I can measure what kind of differences that I would expect. Well, if the difference between these groups is way different, that difference is bigger than what I expect, then I call it statistically significant. And here's an example. This is an example that was out of the book. We've got, we're drilling holes at different speeds. All right, so I'm drilling some holes. One of them I'm drilling really fast, one of them I'm drilling slowly, and then here I'm measuring the distance from the hole where I want the hole. All right, so I have some sort of process, and I can get a lot more done if I drill it quickly. But as you can see, is there a difference in how far off those holes are? Well, like here and here, the distance, this group, this box plot, every single hole that I made with this, and with a faster process, those holes are farther away than what I'd intended than all of the holes that I made when I did it slowly. So this basically, and so you can see there's not even any overlap. I mean, the furthest distance for the slow rate is less than the smallest distance with a fast rate. So this is definitely better. And it's significantly better. Significantly better. Because if I did if I took two samples of things that were identical, like like we were talking about, let's say I did 10 boards at the fast speed, and then I did 10 more boards at the fast speed. I would expect a lot of overlap in those box plots, that they would be, they're not going to look exactly the same, but I would expect them to overlap some, right? So because all of my slow speed holes are closer to where I want them, it's obvious that this is better than this process. So those are statistically significantly better. Okay, that's all I have. Thank you.